Hello everyone. In this presentation, we'll be learning about enzyme catalysis. This diagram is just generalizedly showing you how your enzyme operates. Now this blue color is an enzyme and this is your dark blue which is a substrate. Now substrate binds to an enzyme by your multiple weak covalent bond interactions like hydrophobic interactions, ionic interaction, covalent bonds. Okay, so with these interaction, your substrate is binding to your enzyme to form your enzyme substrate complex, and this enzyme substrate complex is being later broken down to your enzyme and product. This is a very generalized mechanism how your enzyme operates and how is your enzyme converting your substrate into product. Two models have been proposed to show how is your substrate binding to your enzyme, the active site of your enzyme. The first model is a lock and key model, which states that the active site of your enzyme is not going any conformational changes. That is, substrate is exactly fitting to your active site of your enzyme to form your enzyme substrate complex. Okay, so it is just like a lock and key. Okay, so the enzyme is not undergoing any conformational change. The active site is exactly complementary to the substrate. Okay. Now, in second model is an induced fit model, which states that enzyme is a protein, so it can undergo some conformational changes. That is, when your substrate is binding to your enzyme, the enzyme is undergoing some conformational changes to fit your substrate properly. So that it forms an enzyme substrate complex. So in induced fit model, it is showing that enzyme is undergoing conformational changes when your substrate is binding to form your enzyme substrate complex, and the lock and key model state that enzyme is not going any conformational changes. As your substrate binds, it forms an enzyme substrate complex. Induced fit model says that the enzyme is undergoing conformational changes just when your substrate is binding to your enzyme. So these are the two models proposed. How is your enzyme binding to your substrate to form your enzyme substrate complex? Catalytic strategies. Now basically there are three types of catalytic strategies: covalent catalysis, general acid-base catalysis, and iron metal catalysis. Now the enzyme can use one or more types of catalysis to catalyze a reaction. For example, chymotrypsin. Chymotrypsin is a digestive enzyme. Okay, it is a proteolytic enzyme which breaks a protein. Okay, so this chymotrypsin, it uses both covalent catalysis as well as acid-base catalysis to catalyze its reaction. Now, covalent catalysis. In covalent catalysis, there is formation of an reversible covalent bond between enzyme and substrate. There is a temporary covalent bond formed between your enzyme and substrate, and this temporary covalent bond is later broken down and enzyme returns to your original unmodified state when your product is released. Now, enzymes which are undergoing these covalent catalysis usually have nucleophilic and electrophilic centers. Now, which are nucleophilic centers like amino, carboxylic, hydroxyl, imazidol and thiol group? These are different nucleophilic centers. Now, these nucleophilic centers attack your electrophilic centers, leading to the formation of a covalent intermediate. Now, these nucleophilic centers will attack your these nucleophilic centers will attack your different electrophilic centers. Now, electrophilic centers are your phosphoryl group, acyl group, and glycosyl group. Now, now nucleophilic centers will attack your nucleophilic centers which are present on your enzyme, will attack your electrophilic centers which are present on your substrate, leading to the formation of a covalent intermediate, and that covalent intermediate is later being broken down to your product. The example of this covalent catalysis is your chymotrypsin. Now, chymotrypsin is a proteolytic enzyme produced by your pancreas during your digestion. What does this chymotrypsin does? It breaks your peptide bond of the C terminus of the aromatic amino acid. So it is breaking your peptide bond at the C terminus of your aromatic amino acid. So it is what it is. It is just a protease which is breaking your peptide bond at the C terminus of your amino acid. Now this blue color is nothing but your chymotrypsin. This is the active site of your chymotrypsin. In the active site you have your polypeptide chain which has to be broken down. Now what you see different residues of at the active site of your chymotrypsin, histidine, serine, glycine, serine. Now these are the different amino acid residue which are acting upon your polypeptide chain, okay, to break it. Now during this process, what it is, there is a formation of intermediate. A covalent intermediate is formed between your enzyme and substrate, and this is nothing but your covalent intermediate. See, now there is a bond formed between your polypeptide chain and your enzyme, and this is a covalent intermediate. Now this covalent intermediate is later being broken down to form your enzyme and product. Okay. Now coming to your acid-base catalysis. Now what happens in your acid-base catalysis is that your amino acid at the active site of your enzyme are acting as an acid and base to stabilize your transition state. Now see, this is your two reactants. Now these reactants are forming a transient state, but this transition state is not stabilized. So here catalysis is not present, that is your enzyme is not present, so catalysis is not taking place. So now this transition state is unstable and it will back because it is unstable, so it will be converted back to your reactants. Now now in the presence of enzyme, what is happening? Now this is your transition state. Now this transition state is performed in your active site of your enzyme. Now in the active site of your enzyme, you have different amino acid. Now these amino acid will act as either as acid or base, that is they'll accept proton or donate proton based on the pH and microenvironment to stabilize your transition state. And once your transition state is being stabilized, it can be later being broken down to your product. Okay. So what is happening in your acid-base catalysis is that the active site of your enzyme has amino acids. Now these amino acids are acting as an acid or base to stabilize your transition state so that your transition state can be back converted to your product. Okay. Now you can see different amino acid, glutamate, aspartate, right? Either if it is in this form, COH form, it can act as an acid, proton donor. If it is in this form, it can act as base, proton acceptor. So depending on the pH and microenvironment, that is the environment of your active site, different amino acid act as acid and base. So okay, a lysine and arginine, if it is in this form, it can act as an proton donor or acid, or if it is in this form, it can act as a proton acceptor, that is base. Okay. Usually histidine this is which has a pK of 6 under physiological conditions. It is usually acting as, it is present in your active sites of your enzyme, and it is usually acting as an acid base. It is helping in your acid base catalysis. Okay. Now here also you can see chymotrypsin is it also undergoes your acid base catalysis during your breaking of your polypeptide chain. Coming to last one, but it is nothing but your metal ion catalysis. What happens here? You have ions like Mg2+, they interact properly and orient the substrate. Now this is an example. This is your 2-phosphoglycerate. Now this has been oriented properly by Mg2+, so that your enzyme enolase can act upon it and it can be converted into phosphoenol pyruvate. So ions helps in properly orienting the substrate so that reaction can proceed properly. Or sometimes what the ions does, it stabilizes the negative charge. Like DNA polymerase has negative charge. It has phosphorate group which has oxygen which has negative charge. Now DNA has negative charge. The Mg2+ stabilizes the negative charge on your DNA, so that your DNA polymerase, this is an enzyme DNA polymerase which causes polymerization, adds nucleotide and it causes polymerization. That is, it is acting nucleotide and new strand of DNA is being synthesized. So here your Mg2+ ion is helping to stabilize negative charge on your DNA molecules. So here what you can see, your ions, they can either help in orienting your substrate properly, like phosphoenol pyruvate was oriented properly by Mg2+, or ions can help to stabilize your negative charge, or ions can mediate oxidation and reduction reaction. So in this presentation, we saw lock and key model and induced fit model, which shows how enzyme substrate complex is being formed. In lock and key model, we saw that enzyme is not undergoing any conformational change to form your enzyme substrate complex. In induced fit model, we saw that enzyme is undergoing conformational change to form your enzyme substrate complex. Then we saw different catalytic strategies like covalent catalysis, acid-base catalysis, and metal ion catalysis. In covalent catalysis, we saw there are different electrophilic and nucleophilic centers involved in enzyme and substrate to form your intermediate enzyme substrate complex. There is a temporary covalent bond formed between your enzyme and substrate, and later it is being broken to form your product. Then we saw acid-base catalysis, where you saw that at the active site of your active site of your enzyme, different amino acids are acting as acid and base to stabilize your transition state so that your transition state can be broken down to your product. Then we saw metal ion catalysis, in which ions are helping to orient your substrate properly, like in enolase. Your Mg2 plus was helping to orient 2-phosphoglycerate properly. Then we saw in metal ion catalysis that some ions are helping to stabilize the charges, negative charges on your substrate, like in DNA polymerase. Mg2 plus was helping to stabilize the negative charge on your DNA. Then we saw that metal ions can also help in mediation of oxidation and reduction reaction. Thank you very much.